Histone modifications are one part of genetic information that closely relate to gene regulation. Because of this, and the close relationship between histones and gene regulation, a modification or mutation of histones is commonly found in cancer cells. The preliminary work done by Dr. Litt and Dr. Bennett has found that the histone mutation E76K, which is the H2B histone mutated from glutamic acid to lysine at the 76th position, has been found by their lab in mammalian tissue culture systems. However, they were unable to find this same mutation to form malignant cells in Drosophila or fruit flies. So my part in this project was to co-express the H2B mutation with a common oncogene, RASV12, to see if the histone mutation is context dependent in forming malignant transformations. Doing this, I was able to co-express the histone mutation H2BE76K with RASV12 along with IRER deletions to study the malignancies and tumorigenesis in fruit flies. A couple ways that I was able to do this is by studying lethality, counting the flies and seeing the survivorship in percentage between the wild type control and the histone co-expressed with RASV12 mutation. Another way I was able to study the histone mutation was by studying the GFP, which was able to mark hyperplasia, which is a proliferation of the cells and great inflammation of the organs, which signifies cancer and malignant growth. Using the GFP, we were able to study and see the speed of malignant growth and how it differed from the histone mutations and the wild type. And then lastly, a way we studied it was by seeing if the cells were to travel outside of an eye and see if the red eye cell would travel and be seen in other parts of the fly's body, also a great sign of malignancy. Through these studies, I was able to definitely find a greater lethality among the histone mutation co-expressed with the RASV12 and IRER deletion in comparison to the wild type, along with a great level of hyperplasia and malignant transformation and tumorigenesis in the histone mutation co-expressed with RASV12 in comparison to the wild type. In my graphs, you can see that our control, which is the wild type, has much fewer both lethality and hyperplasia in the fruit flies we studied. We were not able to successfully find evidence of the travel of the red eye cell outside of the eye for the fruit fly. However, our two other studies definitely show that there is a great evidence of the histone mutation co-expressed with RASV12 producing great amounts of malignancy and tumorigenesis, and we look forward to studying more the functionality of the genetics that play a role in this mutation and cancer development in fruit flies, and furthermore in mammalian tissue culture cells. Thank you.